Hello everyone, welcome to my channel Pharma Dive. This channel is purely for educational purpose and aims to provide valuable information. So if you are ready to dive into the world of pharma, hit that subscribe button and join the journey. Let's explore, learn and gain together. Today's topic is sterilization indicators used in pharma industry. What is sterilization? Sterilization is a validated process used to destroy or eliminate all viable microorganisms, including bacterial spores, from pharmaceutical products, equipments, components, and manufacturing environments. Importance in the pharmaceutical industry. Sterilization plays a critical role in ensuring patient safety and is essential for manufacture of sterile dosage forms. It prevents microbial contamination and helps maintain product quality, efficacy, and regulatory compliance. Which products require sterility? Various pharmaceutical products require sterility to ensure safety and effectiveness. These include injectable products, ophthalmic preparations, implants, irrigation fluids, and sterile medical devices. Let's understand the need for sterilization monitoring and validation. Sterilization alone is not sufficient unless the process is continuously monitored and scientifically validated to ensure consistent performance. Sterilization monitoring confirms the effectiveness of each sterilization cycle and helps detect any process failure or deviations that may compromise product sterility. It also demonstrates that reproducibility and reliability of the sterilization process under routine operating conditions. Furthermore, monitoring supports routine production activities and ensure compliance with GMP and global regulatory requirements. Sterilization indicators are monitoring tools used to verify whether the required sterilization conditions have been achieved during a sterilization cycle. They provide evidence that critical process parameters necessary for effective sterilization have been attained. Sterilization indicators perform several important functions, including monitoring sterilization exposure, verifying process conditions, detecting cycle abnormalities, supporting validation studies, and assisting in batch documentation. However, it is important to note that sterilization indicators confirm process performance, but not all indicators directly confirm sterility. Therefore, a combination of physical, chemical, and biological indicators is often used to achieve comprehensive sterility assurance in pharmaceutical manufacturing. Classification of indicators. Sterilization indicators are classified into three major categories, which are physical indicators, chemical indicators, and biological indicators. Physical indicators monitor physical cycle conditions such as temperature, pressure, and time during sterilization, typically using temperature charts and recording devices. Chemical indicators indicate exposure to sterilization conditions through a chemical or color change, such as autoclave tape, to verify that the sterilization process has occurred. Biological indicators assess microbial lethality using highly resistant bacterial spores such as spore strips to confirm the effectiveness of the sterilization process. Let's see physical indicators. Physical indicators monitor critical sterilization parameters such as time, temperature, pressure, and humidity or gas concentration depending on the sterilization method. These parameters are recorded using gauges, charts, digital printouts, and thermocouples during every sterilization cycle. They provide real-time monitoring with continuous recording and help in immediate identification of process deviations. However, physical indicators assess equipment performance only and do not directly confirm microbial destruction. Let's see physical indicators used in different sterilization methods. Physical indicators monitor critical process parameters during sterilization to verify that validated operating conditions are achieved. In moist heat or steam sterilization, physical indicators include temperature record charts or digital records, thermocouples for accurate temperature measurement, and pressure gauge or transducer to monitor steam pressure. Modern sterilizers also employ microprocessor control systems for continuous cycle monitoring and documentation, ensuring that validated sterilization conditions are consistently achieved. In dry heat sterilization, physical indicators include master temperature records, probes, or thermocouples for temperature monitoring, and verification of batch temperature settings to ensure effective sterilization conditions. In filtration sterilization, physical indicators include bubble point pressure test for filter integrity verification, determination of pore size and filter performance, and HEPA filter integrity testing like DOP or integrity test to ensure effective sterilization. In gaseous sterilization, physical indicators include temperature monitoring using probes, gas leakage test to ensure gas tight seals, and measurement of gas concentration, pressure, and humidity and exposure conditions to ensure effective sterilization. In radiation sterilization, physical indicators include plastic dosimeters used to measure the absorbed radiation dose during sterilization, ensuring that the required dose is delivered for effective sterilization. Let's see chemical indicators. Chemical indicators use chemical reactions or color changes to indicate exposure to sterilization conditions. Their response is triggered by parameters such as temperature, steam exposure, time, and gas concentration. Common examples include autoclave indicator tape, chemical integrators, indicator strips, Brown's tube, and Roy's sachet, which are used for pack, load, and cycle monitoring. Chemical indicators are classified under ISO 11140 class 1 to 6 and provide rapid visual confirmation of sterilization exposure. However, they offer limited assurance of actual sterility. According to ISO 111401, chemical indicators are classified into 6 classes based on their function and response to sterilization conditions. Class 1 process indicators distinguish process items from unprocessed items, while Class 2 indicators are used for specific equipment performance test. Class 3 indicators respond to a single sterilization parameter, whereas Class 4 indicators respond to two or more critical parameters. Class 5 integrating indicators react to all critical process parameters, and Class 6 emulating indicators provide a cycle-specific response to sterilization conditions. Chemical indicator classes. Class 1 process indicators such as autoclave tape indicate exposure to sterilization cycle but do not confirm sterility. Class 2 specific test indicators such as Bodick test evaluate air removal efficiency and steam penetration in sterilizers. Class 3 and Class 4 indicators monitor one or more multiple sterilization parameters respectively. Class 5 and Class 6 indicators provide higher monitoring sensitivity and show close correlation with actual sterilization performance. Let's see biological indicators. Biological indicators are considered the gold standard for sterility assurance because they use highly resistant bacterial spores to demonstrate microbial lethality during sterilization. Common types include spore strips, self-contained BI system, and biological suspensions. Biological indicators provide the highest level of confidence in sterilization effectiveness. However, their major limitation is the slowest generation of results compared to physical and chemical indicators. Let's see common BI microorganisms. The selection is based on organism resistance towards a specific sterilization method. In steam sterilization, Geobacillus stereothermophilus is used. In dry heat sterilization, Bacillus atrophius is used. In membrane filter 0.22 micrometer, Pseudomonas diminuta, and in membrane filter 0.45 micrometer, Serratia marcescens is used. In gamma or radiation sterilization, Bacillus pumilus is used. In hydrogen peroxide sterilization, Geobacillus stereothermophilus is used. and in ethylene oxide sterilization, Bacillus atrophius is used. Indicators by sterilization method. Indicators vary according to the sterilization method used. Steam sterilization employs physical, chemical, and biological indicators for comprehensive monitoring. Dry heat sterilization commonly uses chemical indicators and biological indicators containing Bacillus atrophius spores. Ethylene oxide sterilization relies on chemical indicators along with biological indicator monitoring to verify sterilization effectiveness. Steam sterilization monitoring strategy. Steam sterilization in autoclaves requires a multi-level monitoring strategy to ensure effective sterilization. Physical monitoring involves tracking temperature, pressure, and exposure time using cycle recorders, SCADA systems, or digital printouts. Chemical monitoring uses autoclave tape, chemical integrators, and Bodic tests to verify exposure and steam penetration. Biological monitoring includes BI challenge studies at worst case location during routine validation and performance qualification. The combined use of physical, chemical, and biological indicators provides robust sterility assurance. Dry heat sterilization depends on critical parameters such as temperature, exposure duration, and uniform air circulation and is commonly used for depyrogenation tunnels and glassware sterilization. Ethylene oxide sterilization requires control of gas concentration, humidity, temperature, and exposure time using chemical strips and biological indicators containing Bacillus atrophius. Hydrogen peroxide vapor sterilization is monitored through H2O2 concentration, contact time, and distribution pattern, with Geobacillus stereothermophilus used as biological indicator. Method-specific monitoring is essential because each sterilization process has a different mechanism of microbial lethality. Let's see regulatory expectations. Regulatory guidelines such as GMP requirements, US FDA expectations, EU GMP Annex 1, ISO 11138, ISO 11140, and USP 1229 establish standards for sterilization monitoring and validation in pharmaceutical manufacturing. These regulations emphasize the use of scientifically justified methods to ensure effective sterilization and product safety. Best practices in pharmaceutical industry include selecting indicators based on the sterilization method, regulatory requirements, and validation needs. Routine monitoring should involve daily checks, scheduled biological indicator testing, and regular calibration reviews. Proper documentation of sterilization records, indicator results, validation reports, and deviation logs is essential for compliance. Personnel should be trained in indicator interpretation, monitoring procedures, and deviation handling while ensuring adherence to ALCOA plus data integrity principles. Conclusion. Sterilization indicators are essential tools for monitoring, validation, and regulatory compliance in pharmaceutical sterile manufacturing. Sterilization indicators provide evidence of process performance. Thank you everyone for your time and attention. I hope this presentation has helped you gain a better understanding of the topic. If you like this presentation, then please like, share and subscribe my channel PharmaDive.